Hello, everyone, and welcome to another exciting Bench Tips webinar. I'm Tanuja Kopal, consulting editor for BioCompare, and today we are actually going to be discussing the excitement around designing and optimizing PROTACs and molecular glues for targeted protein degradation. As you all know, Bench Tip webinars really brings together young talent, postdoctoral fellows, graduate students, to share some of their knowledge, their learnings, their best practices. Sometimes these little tips and tricks get buried in their lab notebooks or in a research paper. And this webinar is really an opportunity for the scientists to come and share some of their learnings and best practices with us, tell us about what worked, what didn't work. And hopefully, talking about all this will help you save some time, costs, and efforts as you are in your labs designing your experiments. So before I get started, I do want to extend a big thank you to our sponsors, BPS Bioscience and Ray Biotech, because without their support, it would not have been possible to bring this webinar to you. So thank you so much for your support. In terms of the format, it's really very simple. We have 3 panelists today. Each of them have prepared like really short talks. So after all the talks are given, there will be time for question and answers. So our goal is to keep things very informal, very interactive. So I will ask you to please use the ask a question button that you see on your screen. So just type in your questions and send it to us, and we will get to it during the Q&A portion of this webinar. But I really recommend not waiting till the end, because we often run out of time and we are not able to get to all the questions. So, type those questions in and send them as you hear the talks. If you're experiencing any technical difficulties, look at the bottom of your screen. You will see a test your connection button. So that's where you can get some help. You will also see some social media widgets. So this is to make it easy for you to share this webinar with people who you think might benefit from attending and listening to what has been shared today. There's also an overview tab that will give you more information about the webinar and about our speakers. There is a resource tab, which I think you should really check out. You will find some useful information that our sponsors have provided, and we also have some papers that our speakers have published. So definitely take a look at that. And there is also a certificate of attendance that you can download if that is something that you need. So we will be beginning our webinar with a talk given by Luis Nieto, Luis is a doctoral student in the laboratory of Dr. Cristina Mayo-Ruiz, and they are at the Institute for Research in Biomedicine in Barcelona. So Luis focuses on understanding the nuances of E3 regulation, and he's working to create some innovative tools to develop monovalent degraders such as molecular glues. So he's going to be giving our first talk. Our second speaker is Dr. Justin Kruit, who is a project lead and the lead structural biologist at the Center for Protein Degradation at Dana-Farber Cancer Institute. So Justin is going to be talking about his work using structure-based design of histidine targeting chemical probes for the covalent modulation of cerebron. And our final speaker today is Dr. Francesca Esther Moriale, who is a postdoctoral researcher in Dr. Tim Clausen's group at the Research Institute of Molecular Pathology in Vienna. And her research focuses on establishing targeted protein degradation in bacteria by reprogramming some of the components of the bacterial protein degradation pathways. So different talks, very interesting talks. So I'm really looking forward to all the talks, but more importantly, I'm also looking forward to the discussion that will follow all the talks. So welcome everybody. And at this time, I'm going to invite Luis to start his presentation. Luis, welcome, and thank you for getting us started today.
Thank you very much, Tanuja, for the very nice introduction. And thank you again for inviting me to this Bench Tip webinar. So in my talk, I will cover molecular glue degraders. I will start with a small introduction, and then I will go through all the process from serendipity to the rational discovery of these kind of compounds. So let's go. Proteins constantly bump with each other in biology, and these protein-protein interactions participate in all physiological processes, including disease. And a number of years ago, it was found that some compounds could orchestrate these protein-protein interactions. These compounds were named as molecular glues. Some examples of these are cyclosporin A, FK506, or rapamycin. But it was especially interesting some molecular glues that could recruit an E3 ubiquitin ligase to a target protein. And those were named as molecular glue degraders. A good example of these ones are the hormone auxin. And this discovery, together with other discoveries with bifunctional molecules, they set the basis of a new paradigm in drug discovery that we know now as targeted protein degradation. So targeted protein degradation is based on small compounds that are called degraders that induce proximity between an E3 ligase and a protein of interest, leading to the ubiquitination and further degradation in the proteasome. So this shift from inhibition of traditional pharmacology to the only requirement of binding has allowed the targeting of proteins that before were considered undruggable. There are main types of degraders, multivalent degraders that are very well represented by proteolysis-targeting chimeras or PROTACs. So PROTACs, they have two warheads, one that binds to the target protein and one that binds to the E3. They are connected by a linker. There are several PROTACs in clinical trials, and they are a very good strategy to degrade a target protein. On the other hand, we have monovalent degraders, smaller compounds, linkerless, that in the case of destabilizers, they bring the protein to a vulnerable state that is further recognized by the proteolytic machinery. On the other hand, we have the molecular glue degraders that, as I said, it will be the topic of the talk, that in a direct way, they induce highly cooperative protein-protein interactions with the E3 and the target protein that leads again to the ubiquitination and further degradation in the proteasome. So a very good example of molecular glue degraders, synthetic molecular glue degraders, is thalidomide that was first released as a sedative in 1957 with the commercial name of Contergan, but it was further withdrawn due to the fetal malformation effects. However, afterwards, the community realized that they had anti-neoplastic activity. So thalidomide, together with the derivatives lenalidomide and pomalidomide, the so-called IMIDs, they were repurposed. But it wouldn't be until 50 years later that the scientific community realized that IMIDs were acting through a molecular glue degrader mechanism. Seminal studies shown that IMIDs were inducing the degradation of transcription factors that otherwise would be undruggable. And some examples are Icarus and Aiolos. Until not so long ago, the only synthetic molecular glue degraders that we had were arisulfonamides and IMIDs. Arisulfonamides were never approved in clinics. IMIDs, they were approved, and they're actually used in a variety of clinical settings. And the main difference between two falls into the affinity of the degrader, the E3 ligase. So for example, IMIDs have a high affinity for cerebron, and this doesn't happen with arisulfonamides, but they kind of compensate it with a large protein-protein interface between DCA15 and RBM39 or 23. So we have learned that molecular glue degraders, they can degrade undruggable proteins. There are smaller compounds, linkerless, that have drug-like properties, and they are a clinical reality. Until 2019 or so, we didn't have rational strategies to discover these compounds. So in our recent publication, we go through all the process from serendipity to the intentional discoveries of molecular glue degraders. And we cluster them in 4 groups, depending on whether we are interested in specific targets or ligases, or we want to do it in an agnostic manner. And I will explain you the rationale of this. For the sake of time, I will just give 2 examples, starting with the target and ligase driven. For this, I will use the last publication of Stuart Stryver, where they aim to find degraders that degrade BRD4 in a VHL-dependent manner. For this, they fuse DNA barcodes to the VHL ligands, and they add a library of connectors, and by combinatorial chemistry, they manage to synthesize, more than 1 million compounds. So after enrichment and further sequencing, they got some hits that could induce a stable ternary complex formation between BRD4 and VHL. And for this, they use several assays. Here I'm just showing one, the nanobit assay. But they also prove BRD4 degradation in a alkaline ring ligase manner, and they even managed to solve the crystal structure of one of their hits. So although this was a proof of concept paper, we really believe that it has so much potential to find molecular glues in a VHL, in a target, sorry, an E3 ligase manner, E3 driven manner. I will go to the next example, that is when we don't know the target or the ligase, meaning agnostic. And we could use this strategy when we are interested in unlocking relevant disease vulnerabilities. So for this, I will use the example of my mentor, Cristina Mayor, that's back in Vienna in the Winters lab. They aim to find the determinants that govern sensitivity to degraders. And they understood that UB2M deficiency was conferring a universal resistance to degraders. UB2M is involved in the methylation process of alkaline ring ligases, and alkaline ring ligases are the largest family of E3 ubiquitin ligases. So methylation is required for these E3 ubiquitin ligases to be active. So by depleting UB2M, they were deactivating more than 200 alkaline ring ligases. They thought of using this model to run a phenotypic screens to find molecular glue degraders in a E3 and target agnostic manner. And for this, the readout would be viability. They used 2,000 compounds and they got 5 hits that were killing the wild type cells, but not the UB2M mutant cells. We keep going with this example to show you how to deconvolute the mechanism of action of a hit that has been found this way in a E3 and target agnostic manner. So for this purpose, we need to know we need to discover what E3 we are recruiting, what target we are degrading, and then we can do interaction studies to see a drug-induced ternary complex formation. So starting with expressing proteomics to find the targets, they saw that they had the stabilization of cyclin K degrader, the stabilization of cyclin K, something that they confirmed by Western blot in a dose resolved manner. So having the target, cyclin K, let's go to the discovery of the E3. So for this, they did genome-wide CRISPR screens, and this was a bit of a special case because they didn't find a substrate receptor. But in the end, they realized that what they were recruiting, it was an adapter, it was DDB1. So having the E3 and having the targets, they performed some interaction studies, and I'm gonna show briefly TR threads, but there are many others. So for this, they fuse a fluorophore to cyclin K and another one to the DB1. And upon drug treatment and increasing concentrations of the target protein, they found that also the signal was increasing, meaning that the hits were inducing proximity between DDB1 and cyclin K. This is the end of my presentation. I just want to give some take-home messages. So we've seen that molecular glue degraders, they are more frequent than we thought, and that we can find them in a rational way. We just need to look for them. And for this, we have an increasing number of strategies that we can use for particular purposes, depending whether we want to find a glue for a specific target or recruiting a specific E3 ligase, or if we want to unlock disease-relevant vulnerabilities, as I mentioned. We have also seen that they have different mechanisms of action, as I show you with the example of DDB1. And also, it's important to mention the importance of a proper deconvolution strategy to discover the mechanism of action of a certain hit. And for this, we have many orthogonal techniques that we can currently use. So I just want to thank my lab, Cristina, and my colleagues and friends, and also collaborators, and also the funders that allow us to perform our research. And with this, I finish my presentation, and I would like to leave you with the next speaker. That would be Justin Kruit. Thank you very much.
Thank you very much, Tanuja, for the introduction, and thank you, Luis, for the nice introduction on targeted protein degradation. So today I'll share the story of our discovery of a covalent cerebron molecular glue degrader. So the imids, such as thalidomide, lenalidomide, and pomalidomide, they all bind to cerebron and act as molecular glues to induce the binding to neosubstrates that aren't normally recognized by cerebron, which then allows the Col4 E3 ligase complex to transfer ubiquitin into that neosubstrate, which leads to its proteasomal degradation. Cerebron is it binds to different substrates, including zinc fingers, kinases, and other enzymes such as GSPT1 by recognizing a structural degron, this beta hairpin loop shown here in purple, which contains no structure, no sequence conservation except for a glycine shown here in yellow at the specific position. And different imid derivatives cause the degradation of different neosubstrates. And so to discover new degradable targets at CPD, one of our methods was to develop a covalent cereblon binder. And these we hypothesized would either inhibit cereblon-dependent degradation or they would induce degradation of new targets. And so we looked at the structure of cereblon bound to lenalidomide and looked for covalently ligandable residues. And we see histidine 353, which is about 3.7 angstroms from lenalidomide bound in the pocket. Histidine has been shown to be ligandable covalently by sulfonyl exchange chemistry. So we designed and had synthesized a number of imid derivatives with either sulfonyl fluoride, sulfonyl triazole, or fluorosulfate warheads. And then the first thing that we wanted to do was confirm that those covalent derivatives bind to the imid binding pocket of cereblon in live cells. And so to do that, we developed a cereblon engagement assay on the similar technology developed by Promega. In our assay, we stably express cereblon bound to nanoluciferase. And then we add to those cells a BODIPI labeled lenalidomide probe. We then titrate in our compounds of interest. And then the luminescence from nanoluciferase acts as the donor, which then activates the BODIPI, and we get a fret signal when no competitor is present. And as we increase the concentration of a competitor molecule, shown here, like such as EM12 control, we see a decrease in that biological fret assay or biological fret signal. And so we ran all of our covalent probes through this assay and confirmed that they do displace the lenalidomide probe, confirming that they do bind the imid binding pocket in cereblon in live cells. We also wanted to confirm that they do covalently label cereblon. So for this, we incubated purified cereblon DDB1 complex with an equimolar concentration of our covalent probes. We incubated overnight, and then we collaborated with the mass spec core here at Dana-Farber, and we showed that there is a nice shift in the intact mass of cereblon when incubated with the sulfonyl fluoride probe, and we see no labeling of BSA or DDB1, confirming that we are specifically covalently labeling cereblon. They also digested the protein and then did peptide mapping to identify the labeling site and confirm that we are specifically covalently labeling histidine 353 in cereblon. And so after we confirm that these compounds are binding cereblon in the pocket in live cells, and they are specifically labeling histidine 353 as designed, we wanted to put them into proteomics and see what if they induce degradation of any new targets. Most of these compounds, we did not see degradation of any targets at all. However, the derivative with the fluorosulfates at position 5, we treated cells, or here we collaborated with the proteomics core at Dana-Farber. They treated cells and then ran quantitative proteomics, looked at fold change versus log p-value. And here you can see with the fluorosulfate, we see potent and very selective degradation of this protein NTAC1. And so now that we've seen degradation of NTAC1, we want to validate that by Western. We also want to validate that it is cereblon dependent. And so we do that by Western blot. And this is actually where we had probably the most difficulty for this project. It was actually difficult to detect NTAC1, endogenous NTAC1 in cells by Western blots with many antibodies that we tested. And what we ended up doing is transiently expressing or overexpressing flag tagged NTAC1 in these cells, and then treating the cells with the compound plus or minus the netylation inhibitor. It didn't, so cereblon and other Col4 or other Cullen E3 ligases require netylation, so binding of the NET8 protein to the complex to be functional. And so if the degradation is a cereblon or Col4 dependent, we should be able to rescue that degradation by treatment with netylation inhibitor. And so I'm showing down here by this Western blot, we confirmed degradation of NTAC1 using either NTAC1 or anti-flag antibody, and then that is rescued with netylation inhibitor, validating that we are seeing cereblon-dependent degradation of NTAC1 with our covalent compound. And then next, we wanted to show a direct interaction between cereblon and NTAC1, and then we also wanted to show that it was the covalent modification of histidine 353 that was mediating that interaction between cereblon and NTAC1. And so we set up a TR-FRET assay that is very similar to our kind of go-to TR-FRET setup. So we purify one protein using a strep tag, and that also contains an ABI tag. So we purify NTAC1 with a strep tag. It's eluted off the strep column with biotin. And then biotin is already in the buffer with the eluted protein. Then we add purified BurA, ATP, and magnesium. And then BurA will site-specifically label that ABI tag. And then we can use europium conjugated streptavidin to have labeled NTAC1. For cereblon DDB1, we purify cereblon DDB1. DDB1 contains a SPI tag. We then separately purify SPI catcher with a single serine to cysteine mutation. We can then site-specifically label that SPI catcher with cysteine reactive malamide Alexa 647. SPI catcher covalently labeled with 647 will then specifically covalently attached to the SPI tag on DDB1, giving us site specifically labeled cereblon DDB1. We then mix those together, concentrations shown down here on the left, and then titrate in our compounds. If they induce an interaction, we'll get a FRET between the europium and the Alexa 647. And so we set up this assay with our covalent compounds, and then read the FRET signal at half an hour, 1 hour, and 4 hours. We see a time-dependent increase in the interaction between cereblon and NTAC1, as indicated by the height of the curves. And then the height of these curves, meaning the interaction between cereblon and DDB1, correlates nicely with the amount of cereblon labeled. at those same time points, strongly suggesting that it is the modification of histidine 353 that is inducing this interaction between the two proteins. So in summary, work here shows that sulfonyl exchange cereblon probes engage the IMiD binding site in live cells. They specifically modify histidine 353. Didn't show today for sake of time, but the covalent probes that did not cause degradation of any new targets do work as cereblon inhibitors for validation of cereblon-dependent degradation. And then we showed that the fluorosulfate IMiD probe induces cereblon-dependent degradation of a new neosubstrate, NTAC1. And so I'd like to acknowledge everyone at CPD that contributed to this project, specifically Lynn Jones, our director, Jeff Dan Silas, who validated the proteomics degradation, Radek, Brianna, and Fidel, who developed the nanobret cereblon engagement assay, Catherine and Rebecca at the proteomics core, and Scott Vaccaro at the mass spec core. And with that, I would like to hand it over to our final speaker, Francesca Esther Moriale. Thank you very much.
All right. Thanks, Tanuja, for the introduction. Thanks also, Justin. And as anticipated, now we change a bit topic and we talk about targeted protein degradation in bacteria. So as you know, when we talk about targeted protein degradation by small molecules, we first look at how the cell deals with protein degradation and then try to hijack these processes with small molecules. So how do bacteria degrade their proteins? In bacteria, there is no ubiquitin. However, some of them have proteasomes. And here you can see a picture of the mycobacterial proteasome recently published. However, most of the bacteria degrade their proteins through these complexes represented here and reviewed in this reference here or in many others. So these proteolytic complexes are formed of an ATP-dependent unfoldase, which you can see here, clip X, clip A, or clip C, that interact with a proteolytic cage that is called clip B. And these complexes normally recognize specific sequences in proteins that are called degrons, but also some post-translational modification. And this is the reason why for the rest of my talk, I will focus on the complex between clip B and clip C. Because when I joined the class and lab, a major focus of research was this post-translational modification, which is a phosphorylation of arginine residues catalyzed by an arginine kinase called MCSP. So arginine phosphorylated substrates are then recognized by the ClipCP complex for degradation. And you can see the recognition sites here that are present on the N-terminal domain of ClipC, which normally recognizes substrates for degradation. And the pockets are highlighted here, and they are conserved in gram-positives and mycobacteria. So this post-translational tagging and degradation machinery reminded us somehow of the ubiquitin proteasome system. And we decided, therefore, to focus on this to find a small molecule that could hijack this pathway. So starting to plan how to design our degraders, the most direct equivalent to eukaryotic products would be to hijack the tagging machinery. So to find bifunctional small molecules, which on one side would bind to our arginine kinase, MCSP, and on the other side would bind to the protein we want to create for phosphorylation and subsequent degradation. However, another option would be to directly find small molecules that bring our protein of interest to ClipCP. And so this would be as if we were designing bifunctional small molecules that direct protein of interest to the eukaryotic proteasome. However, in the case of bacteria, this system is much more simple. And what made the difference here is that while there are no ligands available for MCSP, there are ligands available for the N-terminal domain of ClipC. And that's why we decided to go for ClipC-directed backbone. And also because we can design these bifunctional degraders for two types of bacteria, so for gram-positives and for mycobacteria. So now I will show you in more details how we design this bacterial degrader. So first, we have to select a ligand for ClipC, and as I told you, we already know some ligands, and one of them is exactly phosphoarginine. And you can see here the crystal structure of the N-terminal domain found phosphoarginine. And for mycobacteria, we also know other types of binders that are the cyclic peptide, shown here, among which cyclomarine A, which binds to different pockets on the N-terminal domain and only binds to mycobacteria. Regarding the protein of interest, we decided to start with a simple system because we didn't know if this approach would work at all. So we selected as a model protein first, just monomeric streptavidin binding biotin and a bromo domain binding the compound GQ1, because we have crystal structure of these complexes and we already know where to functionalize these ligands to attach the linker and incorporate them into bifunctional degraders. So here is the tool set of our back products that we synthesized in collaboration with a group of Marcus Kaiser and Uli Katzmaier, and that I will now show into a bit more details. So this is the first back product in which we connect phosphoarginine through a linker to biotin. So the first thing we want to see with this back product is if it mediates ternary complex formation. And what we do normally to see if ternary complex formation happens is to run size exclusion chromatography. And as you might know, if you are working with the multi-subunit E3 ligases, sometimes oligomeric complexes can be tricky to deal with. So since this seminar is also about tip and tricks, one highlight I want to make here is that when we do binding affinity measurements or ternary complex formation measurements, we always start with isolated domains. So in particular, for ClipC, we use the N-terminal domain, which is the part that actually interacts with phosphoarginine. So when we run these proteins together with and without back product, we can see that in the presence of back product, we have a much bigger complex, and this contains both proteins. So indeed, ternary complex is formed. So now the question is, we induce proximity, but do we induce degradation? And the way you normally go when you work with the eukaryotic products is you do Western blot and you see if the protein is degraded. Or for bacteria, there is an extra level of complication, which is often permeability of the molecules that you are using. And another problem can be finding the right antibody for your protein. So there are many more for eukaryotic, for a human cell than there are for bacteria. So we decided to set up an in vitro assay for seeing if there is protein degradation in vitro. And this in vitro assay, we mix together protein components that we purify recombinantly. And here again, since it's about tip and tricks, be careful on the salt concentration, on the pH, and on adding ATP regeneration system. And we then analyze this assay by just running it on a gel and staining it with a Kumasi. So here you can see all the proteins. So I think this assay is very powerful, also because you have internal controls. So you see that in the presence of what happens in the presence of DMSO or different concentrations of the product. And you can see that while the degradation machinery, so ClipC and ClipP, stay stable after 2 hours of incubation, in the presence of 100 micromolar back product, monomeric streptavidin is degraded. So this was a very good result for us because we could, for the first time, demonstrate that indeed we can induce degradation by inducing proximity. And another thing you can see from this assay is that if you see at this lower part of the gel, you can also see how the smaller peptides that form when the protein is degraded. However, here degradation was only observed at 100 micromolar. So one question we started asking is, can we make this protein more degradable? And so can structural features of the protein that we select influence degradability? And in order to start answering this question, I created several fusion proteins between monomeric streptavidin and some proteins that were reported to be substrate of ClipC. And these are shown here with the AlphaFold models. And when I test degradation of these proteins in vitro, I could see that indeed structural features play an important role. And as you can see here, the fusion protein between monomeric streptavidin and CRE was the one degraded more efficiently. And we hypothesized this is due to this unfolded stretch, the determinus that acts as an initiation region. So this is something to keep in mind, that some proteins may be more degradable than others. And what I want to tell you now is also how we use these excellent substrates that we identify for structural studies. Because what I haven't told you so far is that ClipC is active as an hexamer. However, it normally forms an inactive degamer. And this is to avoid uncontrolled proteolysis inside the cell. However, in the presence of a product in our substrate, I observed that degradation occurs. So this means that the protein must be in its active state. So I wanted to use this system to isolate the active state and to do structural investigations because so far there was no structure of an active hexamer of active. So in order to isolate this complex, I wanted to go again to size exclusion chromatography, but this time with a full-length protein. And so this time in the buffer, I have also ATP and an inactive mutant of ClipC, which can bind ATP but cannot hydrolyze it. So as you can see here is that when I don't add back product, ClipC forms, as previously reported, a degamer. These are T classes from negative staining. However, when I add my back product, I would expect a shift of this peak to the right, which means the complex becomes smaller from a degamer to a hexamer. However, what we observed is that the complex was actually becoming bigger, and that running this sample on a gel, both proteins were present in this sample. So when studying these with electron microscopy, we could see that actually this was not an aggregate, but this was something very ordered and was not a single hexamer, but a tetramer of ClipC hexamers. And this was the resulting 3D map, where you can see 4 hexamers coming together to degrade the proteins, held together by these middle domains. And as you might know from cryo-EM, you can focus the analysis differently. So if I focus the analysis on the single hexamer, I can obtain a much better result map. And as you can see here from different orientation, this is the protein in its active state because a substrate is being passed through the ClipC core. Well, let's go back to our proof of concept study. So what I showed you so far was with the Bacillus subtilis proteins. However, we asked the question, how about mycobacteria? Can we also induce targeted protein degradation using the ClipCP complex from mycobacteria? So the answer is yes. When we use our back product 1 using a mycobacterial ClipC1P1P2, we see protein degradation. And this was a very good result for us, because this means that for mycobacteria, we can also replace phosphoargin, because phosphoargin is not a very drug-like molecule. It's also not very stable, and you have to be careful with the pH, because acidic pH is easily hydrolyzed. And also, if you want to store this compound for a long time, you have to be really careful because it's easily hydrolyzed. And the other problem is that it's not cell permeable. So we wanted to do other studies. We wanted to do studies in cells, and that's why we decided to replace it with a cyclic peptide, and in this case is a derivative of cyclomarine A. So we tested the second back product in degradation with the same substrate, and we were very excited to see that also by replacing phosphoargin with the cyclomarine compound, we could still see degradation. So this means that to develop our bacterial product, we don't need to anchor the molecule only to the phosphoargin site, but we can also anchor it to these other sites, so opening more possibilities for these developments. And now briefly, I want to show you how we degraded another protein that I mentioned at the beginning, that is a bromo domain here. So in this case, instead of having biotin, we have the compound GT1. And again, I want to show you how good these assays are, in which we can see degradation of BRTT. We can see the peptide forming in the presence of the back product. And we can also do competition experiments here, for example, with the different head groups of the back products in excess. And in this case, you completely block degradation. So today I don't have time to go in details on the next step, that was testing the degradation inside mycobacteria. But if you want, you can have a look at our paper that is also in the resources. So in summary, I've showed you how we developed targeted protein degradation technology in bacteria, and how these degraded molecules were also useful for structural studies. And as an outlook of this technology, we want to develop degraders that directly target bacterial proteins and to test their antibiotic potential. So this brings me to my acknowledgement. I would like to thank all the people in the class and lab that contributed to this study. This was a huge team effort, which would have not been possible without the contribution of all of these people. And also of our collaborators synthesizing the chemical molecules, our collaborators at Beringer, funding sources, and also here are my contact details. I'm at the end of my postdoc, and I will also be on my group next year. So if you are excited about targeted protein degradation in Bacterial, follow me and stay tuned. I will be also hiring quite soon. And so thanks for your attention, and I hand back to Tanuja for the Q&A questions. I will be happy to take any question.
All right, so that brings us to the end of our talks. Thank you all so much for the talks today, but now we turn it over to the most interactive part of this webinar, which is addressing some of the questions that have come in. So I'm very delighted that we have a guest moderator today with us. Dr. Nikki Kong is the biology group leader at the Center for Protein Degradation at Dana-Farber Cancer Institute. Her group performs phenotypic screens and target validation experiments to discover novel molecular glue degraders. Nikki has received her PhD from UC Berkeley, and she did her postdoctoral training at Harvard Medical School and at Brigham and Women's Hospital. So, Nikki, I'm really glad you could take the time today to join us and also to share some of your insights and ideas on our discussion today. So, for the audience out there, if you have questions for Nikki, please go ahead and send that along as well as those, for our speakers who gave the talks today. Just a quick reminder, the questions have to be typed in the Ask a Question box that you see on your screen. Also wanted to let you know that the webinar will be available on demand in about 24 hours or so, and it will be on BioCompare's website, which is biocompare.com. And all of you will also receive a link to the archive, which will be emailed to you. And don't forget to check out the resource section on our website. There are many resources provided by our sponsors. Our speakers have also provided some of their recent papers that you can download and take a look. And there's also a certificate of attendance. So if you need that, you can download that as well. So with that, I'm now going to turn it over to Nikki to help moderate this live Q&A part of the webinar. Nikki, welcome.
All right. Thank you, Tanuja, and thanks for the introduction. Get the discussion started. So one of the first questions, actually the first couple of questions are about PROTACs. The first one really for any of the speakers. The question is, wondering if when you're designing PROTACs, is there any specific program for this? I'm guessing this is more on the linkerology that we're so familiar with. Are there computational or structural based programs that facilitate the design of PROTAC? Maybe Francesca, since you give the PROTAC talk, you could get the conversation started.
Yeah, although for our products, we didn't use any software, especially because this was something completely new for us. So any information about how the linker should be long or whether we need a cooperative binding or not, this was totally unknown. So the only thing we used were crystal structures, and so information on where you can attach a linker without disrupting the binding. And then went to test that experimentally. So I know there are people trying to do this with softwares and predictions and molecular dynamics, dockings, and so on. But in our case, we didn't use any software.
I haven't.
And
so I haven't done any PROTAC design myself, but I think before I've seen that Mo. The program Mo, you can, if you have a structure of your E3 ligase with your target, you can dock in different linkers to kind of explore the linker between the two ligands. I haven't done that myself, but I think I saw someone show me that you can do that once.
Thanks, Jeff. Another question related to back PROTACs. Wondering if it can work on mammalian cells as well.
I'm sorry, I couldn't hear that. Can you repeat that?
Yeah, so the audience is wondering if the back PROTACs can work on mammalian cells.
So no, the answer is no, because CLIP-C is only present in bacteria. And so that's why the protein can be degraded only in mycobacteria.
Thank you. Another question for Luis. You gave such a great introduction on protein degradation, the field in general. I was wondering if you could expand on the definition of glues, molecular glues versus PROTACs, since that, the Schreiber-Gell screen, the compounds were still rather large. Do they bind BRD4 at the same surface as JQ1? Maybe expand a little bit on that.
Yeah, sure. Thank you for the question. I presented this study as a proof of concept, but it's true that there is affinity for BRD4. So I would say that the main difference between glues and PROTACs is that glues, they don't have affinity for one of the targets. So in this case, it's true that they have affinity for both, and they are more like bifunctional. They have these linkers. But I use it because there are many companies now that are looking for ligands for the E3s, for specific targets, and they use these barcodes. So we really think it has so much potential to link these barcodes to your library of compounds and test stable ternary complex formation and to find these molecular glue degraders. But yeah, in this case, it will be more similar to a PROTAC than to a molecular glue, actually. Yeah.
Thank you. Another question for Justin. So you mentioned that some of the covalent compounds that you found that target cerebron acting as inhibitors. Do you think these can be converted to degraders, either heterobifunctional or molecular glue degraders?
Yeah, I definitely think they can be converted to PROTACs. They bind cerebron very potently, and I think there's plenty of room for adding linkers and attaching to a ligand for some other target. For converting to glues, we did some docking of these compounds into cerebron, and it looks like the covalent linker, the sulfonyl oxygens on the covalent probe would clash with the beta hairpin that's the degron typically degraded by cerebron with IMIDs. So I don't think we would convert it to degraders of other like zinc fingers or typical or like the canonical degron. I think it's definitely possible we could make derivatives and that could maybe degrade other proteins with different degrons, maybe something similar to NTAC1, but we haven't explored derivatizing those inhibitors.
And I guess on a related note, those proteomics volcano plots that you showed, did you look at some of these stabilized proteins that suggest that you're inhibiting cereblon? And are there any candidates in there for cereblon's endogenous substrates? That's kind of still elusive out there.
Looking at the volcano plots, there's one or two proteins that come up a little bit with those inhibitors. None that are common with all the inhibitors. So I don't know if it's like a clue for the endogenous substrate, but we never did try and validate any of the proteins that were potentially stabilized. So yeah, not really any clue for what the endogenous cereblon substrate is, unfortunately. to show they are inhibitors, we just use them to inhibit lenalidomide-induced degradation of IKZF1. And we just looked at that by Western blot.
Thank you for clarifying that. The next question.
Nikki, actually, I had a question, if you don't mind me chiming in.
Yeah,
of course. All of you mentioned like so many different types of assays that you use for various types of studies, whether it was the binding study or target engagement. My question is really, how did you pick which assay to use for? Was that driven by the molecule? Was that driven by the target? I mean, if each of you can just share some light on that, because I'm always curious about how these decisions are made, or is it just trial and error?
I think it usually starts with the question that we want to answer. So for like the cerebron engagement, because we wanted to go into proteomics, we definitely wanted to know that these were engaging cerebron in cells, so we can interpret the proteomics data. I guess if we'd done the proteomics first and we didn't get any degradation, I guess we would have been wondering, well, are these even getting in the cell? Are they even binding cerebron? So I think it usually is question-based, and then whatever assay is best to answer it, easiest and simplest.
Yeah. No, that definitely drives most of the decisions. But yeah, I was just curious. So in your case, yeah, I definitely see that happening. Luis or Francesca, Esther, did you have anything to add to that? And I'm going to ask Nikki the same, because she has a bigger group that she works with.
Yeah. Go for it, Francesca.
Okay, so for us, this was the very first time somebody tried to do something like this in bacteria, so we didn't know if the proof of concept would work at all. And so we went to set up this in vitro assay to remove all this variable about cell permeability. So first we tested it in vitro, because we had this assay that was working for other type of experiments. So that we could answer, yes, it's working. So now we can go in living bacteria and also deal with permeability and all the other things. So this was the main reason for us to go in vitro.
Mm-hmm.
Yeah, no, I just wanted to add that in the end, I believe it's the ideal thing would be to choose several techniques with different readouts to prove kind of the same, to use orthogonal studies to prove the same. So, for example, if you want to test interaction studies, you can do it inside cells. You can do, as Justin said, you can do bread, you can do nanobits, but you can do also in lysate, doing pull down assays, or biotinylation assays. So I think a good way of proving something is just using orthogonal techniques to kind of corroborate the same results. I wouldn't say that maybe for a specific case, yes, but I would say that there is not a perfect technique for the perfect occasion, let's say.
Yeah, no, thank you. Yeah, that's what I was trying to get at. Like, how do you decide? So, yeah, thank you for that comment.
I also echo what everyone has said, and also add that, I went to a seminar recently by Bill Kalin, who really is an advocate for these up assays instead of down assays, which we frequently encountered in protein degradation fields, right? So it's really important to know what led to your signal being down. So, as Luis said, you really need multiple ways of validating whether you're seeing a true degradation or not due to some cytotoxicity effect and, just the signal being disrupted through some other non-target mechanism. So for our group, definitely multiple different ways of addressing, or multiple different assays to address the same question is really important. And also just having the right controls, right? In the colony ring field, there's a lot of inhibitors, and we for every degradation assay we do, for any target validation we use either methylation inhibitors or proteasome inhibitors to make sure that we can rescue that, and also knock out the known E3 ligase if that is known. These are all very important things to keep in mind.
And is someone doing these, testing these hits in vivo then, or is that something not happening as of now?
Sorry, is that a question for the panel?
No, I just wanted to ask you, Nikki, like once your group identifies certain hits for certain targets, are they also being tested in vivo, or is it just all restricted to in vitro studies right now?
No, we definitely have programs that are moving to animal studies. I mean, there are a lot of degrader molecules that are in the clinic now. These are all really well validated modalities. So, yeah.
Okay, great, thank you. No, I really enjoyed hearing all your comments on this topic. It's always at the back of my mind, like how are these how are these assays chosen? So, thanks. No, please go ahead. Do you have any other questions?
I think we have another question about back PROTACs, and I'm also really curious about this. Francesca, what type of resistance, antimicrobial resistance, do you think might arise from back PROTACs?
Yeah, thanks for the question. And this is, I always get this question asked, but the thing is that it's not easy to predict how bacteria will resist because the mechanisms are so different. They can be from efflux, reducing permeability, metabolizing the molecule. But if you're referring specifically to point mutations that disrupt binding, for example, we have seen that since CLIP-C is essential for mycobacteria, then it cannot mutate in the specific site where we had anchored the molecule, at least for what we have seen so far. So there is a lower tendency to develop this point mutation in this specific site. But of course, for each product and each type of approach, we will need to test and look at the frequency of mutations and this type of studies.
That's really interesting. It's definitely something in mammalian E3 field. A lot of companies are trying to discover novel E3s to be leveraged, and some of them, there's definitely a group of thought that we need to target those E3s that are essential to prevent resistance mechanism from arising, right? So cerebron is not an essential E3, but maybe there are others that could be repurposed in this field and potentially lead to less resistance mechanism.
Yeah, this is very interesting. And also for bacteria, in mycobacteria, CLIP-C is essential, but in other bacteria is not, at least when grown in laboratory conditions. So this is something that one would need to check for each bacteria and depending also on the ligand.
I'm wondering if people are using this as antiviral approach as well. Now you have RSV, SARS is out there. Does anyone know of any studies looking at degradation of viral proteins?
I think there are papers. I'm not really familiar with this field.
Yeah, me neither.
Yeah,
because
we come across.
Anyway,
no, but your back protein work, your back PROTAC work definitely raises questions on that, right? Like if you can do it in bacteria, why not in viruses? Why not in fungi or any other?
Because so in viruses, I guess the products that one should develop are for E3 ligases in the cell, because in the end viruses use the cell to replicate. So it would be like hijacking E3 ligases of human cells that are infected with viruses.
And I think there are papers, but I'm not familiar with it.
There are definitely incidents where you can kind of use the viral protein, like known fragments of viral protein that hijack, that basically acts as its own glue to degrade host protein, right? Mammalian protein. Yeah, so I'm sure there's a large effort.
Yeah.
One more question for Luis. Oh, we're almost running out of time. Just really quick, how would you design a non-cell viability-based phenotypic screen to kind of unbiasedly discover glues?
Yeah.
Is there an effort? Yeah.
So I think one example would be the one that Francesca said before, the app assay. It's one example I like a lot. So here you fuse a suicide gene to your target protein. So here you know the target that you want to degrade, and then this suicide gene can, if you treat with a substrate, it can convert it to a toxic compound. So let's say you try your library of compounds, and then those that degrade the fusion protein of DCK with your target, those won't kill your cells, and in parallel you're expressing a GFP protein, so you should be able to detect that fluorescence protein. So this would be one case where there is no cell titer glow or something like that. So, but yeah, there are many cases where that don't depend on cell viability, let's say.
Cool. All right. Well, Nikki, if there are no more questions, I think we can bring an end to the webinar. The hour always goes by so quickly. I'm always amazed. But thank you, Nikki, again for stepping in and helping with the questions and sharing your insights as well. Thank you, Justin, Esther, Luis, for sharing your work and for giving the talks and for commenting. And good luck with everything that you have going on in your lab. I also want to once again thank our sponsors, BPS Science and Ray Biotech, for supporting the webinar, and of course to the team at BioCompare for all the little details that they take care of to make this event possible. So again, quick reminder, an email should be in your inbox giving you a link to the archive. If not, just check biocompare.com and you will have access to the webinar on demand. And the resources are available in the resource tab for download at any time. And for those of you who are looking for certificate of attendance, that's there as well. Once again, thank you all for your time. Thank you for being here. Thank you for interacting with us. And we look forward to seeing you at our next event. Goodbye.